Okay, now let's take a look at the first section of the labs, specifically what materials we are going to be using. First off, we have water, specifically 3 beakers of water, which we will be using throughout this lab.
Distilled water, nonetheless.
Specific. And then we also have our detergent, which we will talk about more later. We also have our baking soda. Again, we will go into that more later. And then we have 2 syringes, one filled with leaf, the other not. And then we have our spinach. Becca, stop eating the material. Moving on. Becca will be going over what exactly the detergent and baking soda solution will be doing.
Okay, so the reasons that we have, I'll bring you guys down a little bit here. The reasons that we have our detergent and our baking soda. So the detergent is made out of dish soap. It's dish soap and water is all that we've got in there. And all of our leaves, our spinach, is covered in a waxy cuticle or a lipid cuticle. And in order to get inside of our cells, to infiltrate them and allow our materials and solutions to get inside of them, to feed them, we need the lipid layer to be gone. We need to get rid of that cuticle. And that is what our detergent's going to do. It'll strip it for us. And then our baking soda will come into play. So as we looked over earlier with our photosynthesis equation, we need carbon dioxide. Baking soda has carbon dioxide in it. And the plant will be able to pull out the carbon dioxide from our baking soda solution and use it the way that it needs to. Now, the other one for our control experiment will be soaking up the detergent, which should break our lipid layer, and water. The water doesn't have anything in it, so it won't be able to feed the plant. So you'll be able to witness if photosynthesis still occurs or not. And the way that we're going to be testing this is the plants. So all of our little leaf discs, you can kind of see them when Raylene showed you that we had these. So we hole punched leaves. We have 10 leaf discs that are going into each of these syringes. And we'll show you how we're going to do this after in just a moment. But we are going to be able to see if these leaf discs float or not. If they float, we know that the leaf disc, this leaf, is performing photosynthesis because, as in our photosynthesis equation, we know that we produce oxygen. And we are going to be filling up all of the little, we call them oxygen vaults inside of our cell with water because we're going to be infiltrating them. We're going to be putting our solution inside of it. So once the plant performs photosynthesis, it will create oxygen again, refilling all of those little oxygen sacs, making the leaf float. You'll have to see what happens. All right, Raylene, you ready to get into our experiment?
Let's do this.
All right. So one of these we have prepared. It already has one of our syringes, has 10 of our little leaf discs in it that we cut out earlier. You can kind of see them hanging out in there. So Raylene's gonna get that one. I will show you guys how to prepare this one. So I need to pull the stopper out and put 10 of my leaf discs inside of the syringe. 3, 4, 5, 6, 7, 8, 9, and 10. Get those all in there. Put the stopper back in, and then in the instructions, make sure those stay inside of there. We want to take as much air out as possible, but we don't want to smash the leaf discs because we still want them to be alive. So we can see Raylene's doing the same thing over there, getting them set.
Just get as close as you can, getting all of the air out without harming the leaves. As you can see in Becca's, she's just barely close enough to the point where it's not going to harm the leaves, but all of that air is out of there.
And now we're going to do, as our directions are telling us, we're following the same lab you guys are. We're going to each draw up a small amount of detergent, about 1 milliliter. So, put our detergent in here. 1 milliliter. All right, there's mine.
And then I'm going to be doing the same. Okay.
And now I need to get baking soda because I am feeding mine. And Raylene gets distilled water. And we're going to draw up another 3 to 5 milliliters of that. We're gonna do 3 right now. So we're both gonna do the same. I'm going to stir up my solution a little bit, make sure that I get some of that baking soda mixture in there. All right, so now we're both hanging out at the same place. And at this point, we're just gonna follow the directions and we are going to tap them a little bit. We're gonna get as much air out of here as possible again. So we just have the solution in. Push right before it starts. You can kind of see what I'm doing here. Right there. I wanna get that just before it starts coming out. All right, and now I just have liquids inside of my syringe. As little air as possible with my leaf discs. And at this point, we need to do the infiltration in order to get everything inside of the cells that we need to. So we are going to create a partial vacuum. We're going to put our finger over the end of this so that no more air will get in. And we're gonna pull the plungers back. Give them a couple little flicks. Get that all mixed in there. And now they should be thoroughly infiltrated. Should be. And then we're going to pour these inside of our beakers. Right then I told you we had 3 beakers of distilled water, so 2 of them are going to be just for where our leaf discs are going to see if they're going to float or not. So Raylan is going to be putting the control into beaker that is labeled 2, backward for you guys. And I'm going to be putting mine into the beaker labeled 2.1. And we are going to come back every minute for 15 minutes, and we're going to observe how many of our discs are floating. But first, we have to get them in there. So, what I'm gonna do is I would put your finger over the end so they don't come out, and then you can just dump them in there. There we are. And you can see right now, stuck at the top. Oh no. You could suck up a little bit of distilled water possibly if they get stuck inside of your syringe. Right now you can see we have no floating. Everything is on the bottom, and that is where we're going to start. We get them?
Nope.
Oh goodness.
A little bit picky.
Well, we will get these guys in the jar. We're gonna cut this video and we will let you guys watch what happens, and then we'll be back.
Hey everyone, so here we have our data from our first trial where we were looking at how our spinach discs were floating. We have our 3 bits of data, the time displayed in minutes from 1 to 15 that we were waiting, our control showing our number of floating spinach discs without baking soda added, and then our treated sample where we had baking soda or sodium bicarbonate added to the water that would mimic the effect of carbon dioxide and cause the discs to float, or so we thought. And a quick look at this data suggests that that is indeed the case. There were 10 total discs there, and we see that at the end of 15 minutes, 9 of them were floating. So now we want to use any kind of graphical software, or you can graph this at home on pen and on paper, basically to display these 2 experiments side by side. So here, Becca will walk you through how to do this in Google Sheets.
So first, It is easiest if we start off by highlighting.
Oop.
Oh, that's not what I was looking for.
Try again.
Yeah, start off by highlighting.
Oh, your keyboard control is different from mine, so you don't need to press twice. Sorry.
Perfect.
There we go.
So once it's highlighted, we can come over to the Insert Chart button. And automatically starts it as a line chart. It's not what we're going for. We're going for a column or a bar, whatever you'd like to call it. And now we need to change our titles, because right now it has it under some funky titles. So we want to go to Chart and Axis Titles. And we can do a vertical axis title first, because you can see we don't have one, and we need one. So this is our number of floating disks. You see that? Put that in there. And our horizontal axis, time in minutes, is good. We can change our chart title from control and treated to leaf disks.
Or photosynthesis experiment, or anything that captures the essence of what your experiment was doing. Leaf discs floating sounds great.
And that's about all that we have to do, because you can see at the top it automatically set in the different colors for us. You can see the control is all in blue, the treated is all in red, and we have all of our axis titles and our chart title.
And ultimately, the point of any kind of data visualization is to show you the contrast quite apparently. So, Becca, would you agree that there was a difference between our controlled and treated disks in terms of floating based on this bar chart?
There was a very large difference based on this.
I would tend to agree. So please make sure that while you're doing these, whether they be on the exam or in this lab itself, that you always label the axes. The one thing that I would suggest that is always necessary is what the unit of measurement is. And so we have our time in minutes here. And on our y-axis, needs a label as well, with number of floating disks. Units of measure are critical. The incrementation is also critical, going from 1 to 15, and this one hopping in twos. So from 0 to 2 to 4 to 6, 8 to 10. And then, of course, label with some sort of key your different colors. Now, it's totally fine to do this in Google Sheets, totally fine to do this in Excel, totally fine to do this on your own paper, totally fine to do it in any other kind of plotting software like R. But it is absolutely critical that you do have some experience doing some data visualization in this class. All right, everyone. So by this point, we've finished our first set of experiments looking at the different steps of photosynthesis, where photosynthesis plants breathe in carbon dioxide and drink in water to produce sugar and oxygen. In our last experiment, we focused mostly on the production of oxygen to watch those discs of spinach float. And they do look pretty cool floating, especially in that time-lapse video, I think. Our second experiment is going to be looking at the second product of photosynthesis, that sugar byproduct. Now, sugar in photosynthesis is produced as glucose, C6H12O6. But the plant doesn't want to store it that way. Glucose dissolves in water, changes the osmolarity of the water, and is not great as a long-term storage mechanism because of that. Instead, you want to store that glucose for a long time as starch, which again is our polymer of that monomer of glucose. Lots of glucose is linked together through the process of dehydration synthesis, will make a giant starch molecule. In order to test whether a leaf has starch, we can use that same macromolecule test that we did in our previous lab, iodine. Now, I'm going to give you two leaves, and you don't know which one has been exposed to sunlight and which one hasn't. One of these two was in the dark for 3 days prior. One of them has been in light the entire time. The one that has been in the dark should have far less reserves of sugar because it wasn't making any.
It was instead depleting its sugar reserves to survive in the darkness. That means that the amount of starch in one of these two leaves should be significantly less. And we'll see that if we can get the dye into the leaf. Just like last time, we had to use a soap, a detergent, to get the cuticle off. We're gonna have to use a couple of different washes to get rid of everything else that's protecting these leaf cells. Both the cuticles, and then the other thing that's gonna be messing with our color pattern visualization, the chlorophylls. In order to do that, what we're gonna do is we're gonna take these two leaves and place them in boiling water for 2 minutes. So I'm gonna start that here. You can keep them kind of separate. So we'll crumple them up. This water is at a nice slow boil. I'm gonna place them in here for about 2 minutes. What's happening here is that this heat is denaturing all of the proteins there. It's stripping the wax off the leaf. It's killing any photosynthesis from continuing to happen because all of the proteins are denaturing. It's starting to break down the chlorophylls that are there. And in that process, you should not damage the starches that are present. The sugar's there, it's there. You're gonna notice that the water that the leaves are boiling in will start to turn slightly green as the chlorophyll pigments pull out of the leaf itself. Now, when we're done with this, what we're going to do after the tissue is sufficiently softened, we will then put both of them into a larger test tube and fill that with a little bit of ethanol. The ethanol will change, again, the chemistry surrounding the proteins to help break down the chlorophylls and the other pigments as well. Because what we're hoping for ultimately is a leaf where everything is broken down except for our starches. So a more white leaf, less color pigmentation. We'll start with this 2-minute hot bath. Swish it around a little bit. You do notice that the colors turn a little bit green here. And then we'll do an ethanol bath in hot water for something like 10 minutes. And the ethanol will pull a lot of those pigments out. After that, we can repeat this step. If you notice that the leaf is not sufficiently white, you can go from the hot water to the hot ethanol back and forth to continue to denature the proteins. But then we're going to need to spread our leaves out on a tray and expose them to the iodine dye and see which of them changes color. If the leaf has a lot of starch, it should change to be dark purple. If the leaf has no starch, it will not change. The iodine will stay brown. And in this way, you should be able to identify which leaf came from the darkroom and which leaf came from the greenhouse. All right, so this has been sitting here now, and you notice that there is a color change to the water. So what I'm gonna do is I'm gonna grab both of these leaves, put them into this beaker, and add some hot ethanol to them. They'll go down to the bottom, and now they will sit here. You'll play the waiting game again for something like another 10 minutes. With all of the waxes broken down, you'll see the light color here, like a tannish color in the water where the leaves were originally. Now with the ethanol added, it should start leaching out the chlorophyll. So when the ethanol has turned a nice green color, you know that the leaves have, the pigment has broken down sufficiently. After that 10 minutes, we'll place them in the dish and stain them with iodine. So I'll see y'all in 10 minutes. All right, so we're not quite through the 10 minutes, but you will notice that this ethanol has gotten quite green. To avoid saturation of the pigment, what I'm going to do is I'm going to switch out the ethanol on these. It should help. So I'm going to just pour out the leaves here and basically just change the ethanol that it's hanging out in. The leaves are getting more white, but they're not quite yet white enough. we can get more pigment out of it, and we will. So we will again leave that maybe for the remaining 4 or 5 minutes, something like that. Keep an eye on its changing color. And after the second wash, the leaves should be white enough to be able to stain with iodine and tell whether they're blue or brown. See you all soon. All right, so here we are after the time frame. It's now a lighter green, quite hot in fact. Pour it out. You will notice that there is a very big difference in the color. The first wash was very, very green. The second wash was a much lighter green. And now let's take our leaves out and see what color they are after all this and spread them out on our dish. You notice that the ethanol hopefully did a fantastic job of draining away all of the color here. That looks fairly well drained to me. And this one looks fairly well drained to me as well. Both leaves look fairly white. Now, in order to find out which one of these was in light for the last 3 days versus which one was dark, and let's hope this works, we're gonna add a little bit of iodine, and what we're gonna look for is a color change to purple. Have to wait a moment for it to infiltrate the cells, because right now it's just sitting on top in an ethanol bath. You can see immediately, once it infiltrates the cells, that this one has purple starch within its leaves. That is definitely changing color. And in the same time frame, we're not seeing purple here. Let's give it another minute here, give it a chance. There's a little bit in some of the veins. You do see a little bit of starch, so it's got some reserves there, but not nearly the reserves of starch of a leaf that is in light for the last 3 days. So from this, I would conclude that this leaf was in light, this leaf has been in the dark. So that hopefully shows you that from our photosynthesis equation, that when a plant is converting this sugar, it's gonna store it as starch over longer periods of time. And in that way, through chemical washes to remove chlorophyll, or through detergents and baking soda to provide carbon dioxide, you should be able to trace each of the individual reactants and each of the individual products through our different stages of photosynthesis.